We have Dr. Brad Hanson. He's a University of California Cooperative Extension Specialist with the UC Davis Department of Plant Sciences. And today he's going to be speaking on branched broomrape, a parasitic weed threat to the tomato industry. So now I'm going to go ahead and pass this over to Brad. So you can go ahead and share your slides.
Okay. Thanks, Cheryl. Thanks, everybody, for coming. Good afternoon. Cheryl already read my slides, so I'll just get right into it. But before I get going, I do want to acknowledge that most of the data that I'm going to present is a graduate student, Matt Bettino, and a postdoc, Pershing Husseini, in my group. And then we've been working quite a lot with cooperative extension specialist Cassandra Sweat, who is a plant pathologist here at UC Davis. So I'll present some data from her group as well. So just by way of introduction, branched broomrapes, more broadly, are a couple of different genera, the Orobonchi and the Philipponchi, that are parasitic plants. They're plants that are parasites of other plants. These are interesting in that they are root parasites, so they attach to the root of the host plant. So you can see that tomato photo on the lower right, where the above ground portion is coming out of the ground, but the plant has no chlorophyll and really has almost no root system. It gets all of its water and nutrients from the host plant. So you see these kind of interesting little yellow spears of the flower stalks there in that photo. There's a lot of broomrapes around. We actually have some native broomrapes in California. Some of the broomrapes have a relatively narrow host range, and others have a quite broad host range. The main problem with parasitic plants, just like other types of parasites, that they can really reduce productivity of the host organism. So in the case of a crop, can really reduce yield or even cause complete crop failure at sufficient density. So broomrapes in California are not totally a new problem. There's been 2 species detected in California: branched broomrape, the Orobonchi ramosa, and there's been, I think, 3 fields now that were reported in California with Egyptian broomrape. That's Orobonchi aegyptiaca. These are the aegyptiaca was in Solano County, so really just a few miles from where I sit at UC Davis. These are a regulatory listed pest by CDFA. Branch broomrape is an A-listed pest, and Egyptian broomrape is Q-listed, which basically means it's probably an A-list, but there's not enough information at this point. So branch broomrape is definitely not new to California. It's been reported on and off since the 40s and 50s. There was a fairly heavy-handed management effort through the 70s and 80s, fumigating fields that had branch broomrape, and kind of got it under control. But it's popped up here again beginning in about 2017. The Egyptian broomrape, the one that I mentioned, has only been reported in 3 fields in the U.S., and those were here in Solano, nearby in Solano County. That has a much broader host range, so it's probably even a greater risk from a crop standpoint, although at this point it appears to have been contained. So I mentioned A-listed and Q-listed rather. So the A-listed pests here, I've got the definition from the CDFA website, but essentially these are pests that are subject to strict quarantine and oftentimes eradication efforts because they're new pests. So we're trying to keep them under control, containing them and eradicating where possible. So essentially, when a field is reported with a pest like branch broomrape, an A-listed pest, it's quarantined and can't be harvested. And there's a procedure that happens where the field is held, and only non-host plants can be grown for some number of years. As I said a moment ago, Q-listed is basically suspected to be A-listed, but the status is not, there's not adequate information to make that final determination. So functionally, they're fairly similar from a grower perspective. Just a quick primer, I guess, on broomrape life cycle. So if we start in the right-hand side of this photo with the seed production, the seed. So the seed are in the soil. These are obligate parasites. I think I forgot to mention that, which means that the plant can only germinate when there's a suitable host. So the seed is dormant and doesn't get it gets a chemical signal from the host plant. So essentially the seed, quote unquote, knows that there's a host plant nearby. These are tiny seeds, which means they don't have a lot of energy. So they can only germinate when they're relatively near a host plant root. They germinate, they start to make a small root that grows towards and encounters the host root, at which point it makes an attachment and forms a structure called a turbuckle. And this is all happening underground. And essentially this turbuckle is a reservoir for storing nutrients and water. And they can be a fairly large mass eventually, because they, as I said earlier, they take all of their resources from that host plant. Fairly late in the life cycle of probably 3/4 of the way through the life cycle of this plant, it starts to produce those shoots that you see in the upper left corner that emerge from the soil surface. And then they flower in relatively short order. The time between the emergence of the first shoots and the first flowers is likely less than a week or two in most cases. So it's hard to detect because most of its life cycle is underground. Then interestingly, they produce a lot of seed. You see the purple flowers in the kind of the top center. Each of those flowers will make a capsule, and each capsule, you can see the seed production in the upper right. Each capsule will have dozens to hundreds of seed. So that relatively small statured plant that you see in the just left of top dead center, those spears are probably no more than 8 inches tall. There are likely hundreds of thousands of seed being produced by that little clump of broomrape. And interestingly, from a biological perspective, those seeds are really durable and can last a long time in the soil, probably decades at least. So once you have a problem like this, the seed are likely in the seed bank for a long time, awaiting a suitable host to be grown in that field. Just a little bit of an idea what the plant looks like. These are some field photos from a research site that we have been working in for a few years. On the left is a broomrape attached to a volunteer tomato seedling. And then on the left, you see a more typical clump of broomrape just starting to emerge from the soil surface from a transplanted tomato, and that Matt's holding in his hands there. These are some photos from some research projects we've been working with cooperators in Chile. So we haven't seen this level of density in California, but this is what it can look like. So in the upper left, you see Matt holding a clump of broomrape attached to that horizontal tomato root. And then we've dug up a fairly large clump in that right-hand photo. All of those plants, all of those spears or stalks of broomrape are growing on the same tomato root system. So you can imagine the amount of resources that are being drawn from that host plant by having that much parasitic plant material growing on it. And then, as I mentioned before, the seed are numerous, so that Matt, that hand there, he's just given a little shape to that mature broomrape stalk, and very likely he's got several 100,000 seeds that have come out of that plant. The photo on the left, you can see relatively high densities of broomrape in that processing tomato field. Again, those are in Chile. So I'm going to start getting into some research projects here, but this is, as I said, an A-listed noxious weed in California. We don't have solid management plans for this parasitic plant currently. This is a matter of scouting, reporting, quarantine, and crop destruct in those reported fields. So the second bullet there is slightly out of date. I said we don't currently have data on suppression and control with registered pesticides, which is not quite true. There's a new pesticide registered as of 2023 that I'll get into and talk about based on this research. But so far, both of these species have been detected in conventional processing tomato fields. So that suggests that we really don't have adequate control with our currently registered herbicides. Quarantine treatments are basically based on soil fumigation, primarily with methyl bromide, which is, horrifically costly, regulatorily challenging, and environmentally challenging as well. So fumigation was used pretty heavily in the 60s, 70s, and into the 80s, but is not going to be used much more than on a fairly limited basis with our current issues. So this takes me to the point of saying that minimizing the spread is really, really important. And I'm going to get into some research on equipment sanitation in a few minutes. But if you're a tomato grower and you don't have this problem, I would recommend doing your very best to make sure that you don't get it because we don't have good management programs, and it's a quarantine pest. So detection is really where the threshold is at, not even getting into the yield reduction levels. So the first set of data that I'm going to talk about is helping develop mitigation approaches, herbicide approaches for management should this ever become a non-regulated pest. I've got a photo from researchers in Israel. This is I guess I didn't mention this, but the broomrapes are native to the Mediterranean region. So, Mediterranean climates like California, but in particular, our colleagues in Israel have been working on this for several decades. We've had some cooperation with researchers from Turkey, Egypt. There's a lot of the Mediterranean climate areas of the world have this broomrape as well as other types of broomrape. And Chile, I mentioned we're doing work with Chile. So the Israelis have developed what they call this PICKET system. PICKET's an acronym that makes more sense in Hebrew than it does in English, so we'll just leave it at PICKET. But essentially, they've got chemigation treatment. So they have a couple of sequential applications of herbicides that are targeting specific broomrape life cycles. And they're getting really good control, not perfect control. If you look around in the other parts of this image, you can see a broomrape plant scattered here and there, but relative to the non-treated control in the foreground, they've got very workable solutions. It is not a quarantine pest in Israel, so it's just another weed that they're managing there. So we've been working for a couple of years to kind of adapt the Israeli PICKET system to California. The primary limitation was that the herbicides used in Israel are not registered for use in tomato in California. So for the first couple of years in 2019 and 20, we were looking at herbicides called imazapic, which is a rangeland or peanut herbicide in the US that is not registered at all in California, and a PPI treatment of sulfosulfuron, which is a wheat herbicide in the US, or a bare ground, vegetation management herbicide that is registered in California, but not in tomato. In 2021 and 2022, we switched from the imazapic to imazamox, primarily because it appeared to be insurmountable barriers to registering imazapic in California. So we're trying to find a sister compound in that same chemical family. And again, pairing that with the sulfosulfuron. Excuse me. In 2022, we continued some of that work, but we started looking at chemigated rim sulfuron in a more intensive way. The Israelis have settled on imazapic as their main herbicide in that class, but other researchers like in Turkey, Italy, I think in a couple of other Southern European countries have been using rim sulfuron, which we have as matrix that's already registered in tomato in California, just not as a chemigation treatment. So in 2023, we were fortunate to help the CTRI, the California Tomato Research Institute, champion a 24C special local needs label for chemigated rim sulfuron, and that was available for use in 2023. So we started looking at that in a more systematic way to help, fine-tune that protocol over time. In addition to those chemigation treatments, we looked at some foliar applications of a product called malic hydrazide, which is a sprout inhibitor in potatoes and onions, but also has some activity in this arena. And then we've done just a little bit of tomato variety screening, both in the quarantine greenhouse and in field trials. So I'm not going to go super data heavy here, but I want to give you some sense of what this problem looks like. This is a infested grower site in Yolo County. I should have also mentioned this, that the branch broomrape primarily has been reported and a major problem in Yolo and surrounding counties. But I would say there's really no barrier that I am aware of that would stop this plant from growing anywhere we grow processing tomatoes. So it's currently somewhat limited, but I think, I have some concerns that it could become a statewide problem. So in the upper left, this is work from Matt's PhD work. The upper left shows kind of just a sense of this field. We've got a great cooperator who's been working with us in spite of having this quarantine pest. He's still been farming tomatoes on this quarantine site, and then we destroy them without harvest just so we can do this research. So I can't emphasize enough how important having grower cooperators are for any of our UC research trials, but particularly for really challenging ones like this Broomrape. In the center photo, you can see 3 or 4 rows of tomatoes. In this particular experiment, the plots are the whole row. So end to end, these are 120 feet long. And you can see 3 rows there with a lot of flags in the left and middle row. And the right-hand row just has a handful of flags. So we are making some progress with some of the chemigation treatments. And I feel really, really positive about where we're getting. The photo on the right I put in here just to remind you that our primary crop host of branch broomrape in California is tomato, but it actually can grow on related species like that nightshade plant that's in somebody's hand, my hand possibly. So some of the related solanums are I see somebody in the chat said they can't see the photos. I hope that's not true for everybody, because this will be a very boring presentation. So anyway, just to make a point that the broomrapes can grow on other species, and a couple of these weed hosts that are closely related to tomatoes are clearly hosts, as you can see in the right-hand photo. I've got just a little bit of data from that trial, from the trial in 2022, and basically the tall bars are bad. That means number of broomrape plants per 100-foot plot. So you can see in the non-treated, the checks on the right, and a couple of other treatments kind of on the left, we're getting 50 or 60 broomrape clusters per 100-foot plot. I circled 3 of the treatments here. That's the rimsulfuron chemigation, rimsulfuron with sulfosulfuron PPI, and then the sulfosulfuron imazapic with the blue circles. So the blue circle treatment is the Israeli standard. And the red circles are the ones that we've been pursuing a little bit more thoroughly, particularly after getting that 24C registration. This is a bit of a good news, bad news slide, I think. So what I see with the blue circle is that what the Israelis are using is reducing our broom rate by 75 or 80%, which isn't as good as I'd like to see it. But on the bright side, our rimsulfuron treatments are doing just about as well in our system as the Israeli standard. So this gives me some hope that we can work on the protocol, fine-tuning timings, and application windows and so forth, and do a better job. So this is not really workable while this is a quarantine pest, because even these plots with 80% reduction would still be quarantined because the broomrape is present. But it makes me hopeful that we've got some management tools at our disposal if that becomes a weed that we just manage down the road. I think that's all I wanted to mention here. Well, I will say on the very far right, you see miscellaneous varieties. We had a couple of early stage variety trials in this particular year, and most of those looked pretty good. So I think there might be some genetic solutions, potential for genetic solutions out there, and I'll talk about that in just a couple of minutes. I'll give you just a little bit of data from our cooperators in Chile, the UC Davis Life Science Innovation Center, Chile Life Science Innovation Center rather, Juan Carlos and his team. Basically, he was doing the same trial in Chile that we then did later in the year in California. So this was a 2022 data set that preceded actually the data set that I just showed you. They harvest in about February or so, just as we're getting ready to start planting. And I would just say here, treatment number 4 and number 8 are the chemigated rimsulfuron treatments. Treatment number 1 and 11 were non-treated. So they were getting, reasonable reduction in Chile as well, although maybe not quite as good. In 2022, they were using drip tapes on the soil surface, and while we were injecting chemicals through the buried drips. I think that might have been the main reason for their somewhat reduced efficacy. You can see the small figure on the lower right. The Chilean group was also looking at some planting dates. We had planned to do that in California also, but both in 2022 and 2023, our weather windows precluded our really early planting. Well, actually, one year we had a problem with the late planting, one year we had a problem with the early planting, so we don't have these data. But there does seem to be some mixed results, I would say, depending on planting dates. I think that one's still open for discussion, whether there's benefit to pushing your planting later if you have an infested field or earlier. We're still working on that. So I want to talk just a little bit more deeply about the field data from this year, from 2023. As I mentioned, we're largely targeting the chemigated rimsulfuron application timing and alone and paired with the sulfosulfuron. This is the year we had the malic hydrazide as well, which is a hormone, a plant growth regulator, PGR. In this particular year, we worked with Ag Seeds to look at 5 of the really common varieties at kind of a grower scale to see if there's just happens to be anything different in terms of sensitivity to broomrape. Let's see. So the main trial was HM 58841, and you can see the details there. The varieties are on the lower bullet. There was a couple of other HM varieties and then a daughter-resistant variety. Some question about whether the resistance to daughter would also impart resistance to broomrape. They are not related, but they are both parasites. But so that's a little bit of a shotgun screening. So this figure is much like the one I showed you earlier. Tall bars are bad. That means there's a lot of broomrape in a 120-foot row, 120-foot plot. Short bars are good. So we'll just run across here. Basically, the controls had 20 to 25 clusters per 100 feet a row. Pretty much all of our chemigation treatments looked pretty good. I won't belabor these too much, but there's some differences in rimsulfuron applied 4 times at 1 ounce versus the protocol of 3 times at 1.33 ounces. GDD is a growing degree day model versus the days after transplanting model. That's the DAT. The malic hydrazide was 3 foliar treatments with a couple of different rate structures. And I would say on the very far right to this, you see Amaza-Pic, which is the Israeli standard, as well as Amaza-Mox that we looked at in those first couple of years. Those bars look really good from a broomrape standpoint. Unfortunately, they also look really bad for tomatoes. We had very severe crop injury from both of those herbicides in this particular year. So I think the low broomrape is really a function of severe tomato injury, which further kind of emphasizes our decision to stop chasing the Amaza-Mox protocol. So again, we had pretty positive results with most of these rimsulfuron treatments. Let me see here. The very lowest numerically, I think you can see my cursor here. Sulfosulfuron rim 3 times 1.33. This is essentially what the well, actually these 2 are what the 24C registration allows. So we've got a very good reduction, 85% reduction in broomrape with that protocol. So I think that we've got something to work with here. So I've mentioned a couple of times this rimsulfuron 24C, that's the special local needs label. CTRI supported this label. Corteva, who's the registrant of rimsulfuron, was very, very supportive, finding data from their European arm to help get the data to DPR to support this label. Very, cooperative effort with CTRI, Corteva, CDPR, as well as the group in the UC system. So this was registered in, I think about, well, let's see, I think it says on here, last fall of 2022. So the label was available for use in spring. And I think this was used quite widely, particularly in Yolo County in 2023. I'm just going to go through this very briefly to give a sense of what this special local needs. This is basically, so tomato growers are already using Matrix very commonly as a pre-plant herbicide, as a foliar herbicide. A lot of tomatoes are already treated with Matrix. However, this 24C allows this subsurface drip application. So the herbicides are put into the irrigation system, into the buried drip, and chemigated on with irrigation water. The idea being you're putting the herbicide in the tomato root zone, right where the broomrape is germinating, and at a time, at 3 application timings, trying to target that germination and early attachment so you can get control of that parasite before it becomes established. The protocol you can see here, this is 1.33 ounces of product per application per acre, and through the subsurface drip only in California. You see here, total of 3 applications are that the label requires 3 applications of 1.33 ounces, beginning just a few days after transplanting, and then a 10 to 15 day interval. So that 1.33 times 3 applications is 4 total ounces, which is your maximum yearly matrix allowable in a single tomato crop. So the restrictions: don't make more than 3 applications in a year. Do not apply more than 4 ounces total, because that's the yearly max on the existing matrix label. One of the big challenges is if you use matrix using this chemigation protocol and you use all of your 4-ounce annual allowable use of the herbicide, you cannot also use matrix for foliar, for control of other weeds, basically. And then number 5, I just want to emphasize here, if you're using this herbicide for broomrape control, this use pattern really isn't going to give you much help for anything besides broomrape, because you're putting that herbicide, 6 or 8 or 10 inches below the soil surface, which is where you need it for broomrape, but that's not going to help you with your grass weeds and your nightshade and so forth to any appreciable degree. So this is really a broomrape only protocol. I'm not going to go through this. This basically just says all the stuff that I mentioned earlier, but I wanted it in the slide set. The bullet points are the important ones that I already said. So 1.33 ounces 3 times in a season, beginning 30 to 70 days after transplanting. These relatively, the windows here are something we're working on in our research to try to figure out if that's the best, or if we can improve our control with maybe starting earlier or going later. We're trying to understand broomrape in California. A lot of what we're doing is based on Israel and Turkey, which isn't exactly the same system. Here down on the bottom, you see, again, I'll emphasize, do not apply more than 4 ounces of matrix per year, and do not make more than 3 applications in a year. And the same 45-day PHI still here. This is the same as it was for foliar matrix. So this doesn't really change anything. And for broomrape, you should be using this well before this time frame anyway. Okay, I'm going to switch gears just a little bit and talk about some of the other research that my group is doing. This is work that Pershang that I mentioned. I'll make one quick aside here. So working with a quarantine pest is new on me. I'm generally a fairly regular weed researcher. With quarantine, even though we've got this weed in the field, we can't work with it in our regular greenhouse. We have to work in it in a biosecurity 3 greenhouse on campus, which is a shower in, shower out facility to minimize the risk of a quarantine pest like branch broomrape or any of the dozens of horrific insect pests that they're working with in the quarantine greenhouse. So it's a pretty neat facility. I think it's the only biosecurity 3 greenhouse in the Western US, or certainly the only one in California. So it's fortunate that we have that available. But a lot of what Pershang is doing in the greenhouse is the more biological modeling. You can see, looking at seed production and emergence biology to make sure we're getting our timings reasonable, looking at differences between branched and Egyptian broomrape, as well as some other broomrapes. And then we've done just a little bit of work with different tomato cultivars or chemical evaluations. The photo on the right just shows herbicide injured tomato plants. This was one of our early projects where we were injecting herbicides at the same time, trying to look at crop safety. So you can see broomrape growing in some of these plants, and some of those tomatoes look pretty sad from the herbicide research projects. Other types of things that Pershang is doing, glass-fronted rhizotrons, so we can actually look at the tomato root, as you see in the left-hand photo here. That's a broomrape plant, a tubercle, and just starting to elongate. On the right, I think that's a close-up of that same one. You can see that underground attachment point, and then these are the beginning of the stalks that are working their way towards the soil surface. You see a few here in the upper right portion of that photo that are already emerged, and these ones up against the glass will be probably emerged in a day or so from this photo. This is useful for helping us kind of time our application windows for our camigation treatments. And then more recently, Prashang has been working on alternate hosts. In her hands there in the upper right photo, you can see that's a tomato plant, obviously, but these small cups have been really useful. You can see the early attachment, so we can get a better idea of exactly when the attachments are occurring on tomato or any other host plant. And then kind of exciting, I would say, in the lower right, we're working with a early stage development of a germination stimulation chemical. And Prashang has been working with this in Petri dishes. We compare it to this GR24, which is our lab standard for stimulating germination. But these other two compounds were developed for stimulating germination of striga, which is a parasite of corn and sorghum and other grass and millet. It appears to be quite good at stimulating germination of broomrape as well. She's right now at the stage where we're looking at it in soil. The idea being you put this germination stimulating chemical in the soil environment. The broomrape thinks there's quote unquote thinks, I don't want to personify this too much, but the broomrape gets a signal that there's a host nearby when there is not an actual host. So the seed germinates, doesn't find a host, and then the seedling dies. So we're looking at this in soil in the greenhouse right now. My hope is we'll be able to take this to the field next year or the year after to see how this works in the field. The researchers in Saudi Arabia, where this was developed, they're actually working with this in Africa for striga control, and they're doing field tests right. I think they have been for about a year. So this is pretty exciting, although very, very early stage, probably quite a number of years from being something we could use in California, but very promising nonetheless. One of the main things that Prashang has been doing in the CRF, the Contained Research Facility, or the quarantine greenhouse, is looking at different quaternary ammonium compounds as a way to sanitize or kill seeds on equipment. And I won't belabor these data too much, other than there's a bunch of different compounds, the quaternary ammonia or quats. You probably heard that name. What we found in Petri dishes, many of these quaternary ammoniums are quite effective, even at quite low doses. Like you see that bottom figure here, the didecyl dimethyl ammonium bromide. Any of the doses we tested at any exposure time from 1 to 10 minutes, all were very good at inhibiting germination or killing seeds, reducing viability of those seeds. So that was very positive. I will say, check with the local ag commissioner about the compounds that you're thinking about using to make sure that they approve it for that use. A lot of these quaternary ammonium compounds, they're widely used in sanitizing medical facilities, food facilities, but they weren't developed for broomrape management on farm equipment. So they don't specify that on the label, but your local ag commissioner can give you the information on whether they would approve that for that use. So let me back up. I would say this is the good news slide. This is the bad news slide. So Prashang has been looking, as part of the project I'll mention here in a moment, we've been looking at QAC, or quaternary ammonium compound activity in the soil environment. So in each of these figures, or each section of this figure, there's a different quaternary ammonium. This is Mg4 quat, this is flow quat, and this is chem quat something. I don't remember what the CQT is, but the 0 means it was in water. And then 10, 30, and 50 is there's a little bit of soil debris added to that mix. Quaternary ammonium are cationic, and they're relatively quickly inactivated or deactivated by soil. You can see in each case here, the efficacy, broomrape mortality was greatly reduced in the presence of the soil. So this leads into the sanitizer project that I'm going to talk about in just a moment. Oh, see, yeah, I had a break there. So I mentioned Dr. Cassandra Sweat at my introduction, and I'll come. A lot of what I'm going to show here is work that she's either done or that we're heavily involved in. Dr. Sweat is a plant pathologist who does a lot of work on diseases like fusarium and bacterial, diseases of various sorts of tomatoes and other vegetable crops. It's really, minimizing the spread of any of these soil-borne pests or pathogens is really, really important. It's even more important for a pest that you don't have widely spread, like branch broomrape or fusarium race 4 or something like that. So we've been doing this project for a couple of years now with largely with funding from the CTRI, the California Tomato Research Institute, and more recently with funding from the CDFA Specialty Crop Program. So Cassandra's group is doing the plant pathology group part. My team is doing, Pershang primarily is doing the broomrape work, and then CTRI Director Zach Bagley is involved in this as well. So I want to talk about what we called equipment sanitation or Project Clean Machine. We were trying to come up with something catchy. We started working primarily initially with harvesters, thinking that those are a very likely route of spread from field to field and from region to region. And if you're a tomato person, you know that these are huge machines, they're complex, they're convoluted, and they're dirty, and they're hard to clean. So we've been doing some work in recent years. These photos are actually just, well, the photo in the upper right is just from last week, where we were doing post-harvest sanitation work with a local grower. The lower photo is a little earlier in the season, different machines. You can see, the amount of tomato debris and plant debris, like in that center photo on the dirt sort belts, and these things are dirty, they're covered with goop. And so on parts of the machine, it's mostly plant debris, like that center photo. But of course, on the undercarriage and the tires and the wheels, that's mostly going to be soil debris. And if you're thinking about the risk of spreading a seed, like branch broomrape seed, you're probably thinking more about soil and mud and dirt. If you're thinking about spreading diseases that might be more in plant debris, like fusarium or bacterial pathogens of various sorts, probably the more plant debris laden areas are going to be of greater risk. So we've been working together trying to accomplish two things here to minimize the spread of excuse me, of pathogens as well as branch broomrape seed. So basically, I'm not going to go too deeply into this slide, but this is really important for a lot of emerging pests. Branch broomrape is a primary concern right now because that's front and center in front of us. The distribution mostly is limited to the Yolo County region, mostly Yolo County and a few reports in nearby counties. But the concern is that harvesters, trucks, and trailers are potential vectors for long distance transport throughout the tomato industry in California. So what we're trying to do is help growers and processors develop best management practices to sanitize farm equipment. This is not something that's that has really been done in the recent past. I mentioned the QUAT labels earlier. So this will also help generate data to help amend those labels for getting broomrape specifically, or for this use pattern specifically on those sanitizer products. I've also got a photo here of nematodes. This beginning next year, we're bringing in a nematologist because there are some soil-borne plant parasitic nematodes that are real important as well. So while we're doing all this sanitation work, we want to make sure we get the most bang for our buck, so to speak. This is some of Dr. Sweat's data, early evaluations of types of equipment. Basically, how dirty do these pieces of equipment get? In this case, they used a 1 through 4 scale, and the horizontal line here, the one line is clean, basically didn't look like it had any debris. But if you look, the harvesters seem to be both the dirtiest, and most difficult to clean. Transplanters, trailers, these things are all loaded with different types of debris and move considerable distances. She also looked at some other types of equipment, the tractors themselves, things like vine diverters, cultivators, using microbial loads as a bioindicator. So the numbers don't matter so much here, but basically the tall bars are bad. That means they're more more dirt and debris and microbial activity, more microbial life, which would suggest that more likely for broomrape seed to be surviving in that area as well. And again, we focused on the harvesters because they're so complicated, complex, large, and difficult to clean. In the first year, this is something I thought was really interesting. So if you look at this figure, again, tall bars are bad. That means they have the most microbial propagules. So T0 was ×0 before we did anything. So Cassandra's group went through and took swabs all over the machines before any washing step was done. The first step was just to use compressed air. And you can see, and I thought it was really impressive, both the compressed air or air plus high pressure water, a power washer. Just using air, you're removing debris, right? So if you go through and you knock all the big chunks of soil off, you get the air compressor out and you blow all the loose soil and debris off. And then particularly if you follow the power washer, you've got a 80 to 90% reduction in microbial loads just by the physical cleaning. If you start adding sanitizers like this MG, let's see, no, I'll come back to that in just a minute, but I think that's just the first take home is get the loose big chunks of debris off with air and possibly also with water. You're making really good progress over doing nothing, which is what most of the industry was doing prior to this. So we wanted to add to this the quaternary ammoniums. I mentioned these earlier, the FlowQuat, ChemQuat, the MG4 So working to understand how best to use these on equipment and to get these target pests added to those labels, which don't typically focus on harvest pests. They think about food safety issues largely, or medical sanitation. Okay, again, starting with the harvesters. So I showed you, I think you saw some of these data in a little different bar chart earlier, but we've got good data on quaternary ammoniums on broomrape, and that's this left-hand photo. So some of these are quite good at low dose, at low concentrations. Fortunately, the quats that are good on broomrape are also good on, at low doses, short exposures in clean environments. When Cassandra looked at those same products on fusarium, fortunately, those same products are also very good on fusarium. We had some worry that the products that would be good on one class might not be good on the other, but it looks like if we can control broomrape, we can do a good job controlling other pests as well, other pests of quarantine, of high risk. So I showed you the first part of this figure earlier, but as we add those quaternary ammoniums, whether they're MG4, whether they're the ChemQuat, we did some work with a foam agent to help increase retention time on vertical surfaces in particular. So, if we reduce air and water, take you down 90%, add the quats to that, you're taking, getting close to 97, 98% control. So I think there are good ways to reduce the risk. Difficult to get no risk, but a lot of what we do is risk reduction. So I mentioned the foaming, that's a photo of a field day we did on campus last year. The foam helps with the cleaning, but mostly it helps keep the surface wet and keeps the quaternary ammoniums in contact with that surface longer. And you can see here the green bars are just the QAC. The yellow bars, they don't exist because they were very effective, but that's a QAC plus foam. So that worked really well, particularly on some of those difficult to clean areas like the suction duct, which is this horizontal tubing where the plant debris is blown out. Take home message, QACs work, but the efficacy varied somewhat by location, mostly because of the difficulties in effective cleaning. So you can see some of the undercarriage, like the center photo looks like it's kind of the axle in the chipper area of that harvester. It's difficult to clean. It's a wet, muddy job. It's hard to get under there. So that's a challenge. But the more easily accessed areas like the belts and chains and so on are pretty easy to clean. You can do a good job there. So cleaning is hard. And so a lot of what we've done in the last year or so is helping get at how to do that better and how clean is clean. So, spending hours cleaning the machine between fields is the best way to do it, but it's not very feasible during the fast-paced harvest season. So a lot of what we're doing is trying to figure out how to streamline that and figure out what the threshold really needs to be. So I'm going to try to wrap this up because I know I want to get to the time for questions in your quiz here, but I do have a couple of links I'll share. So we've got the sanitation best management guide that you can, it's a living document. We're updating this as we generate new data, but this is available. But basically this says you've got to clean the machine before you use the QAC. If you spray QACs on dirt and debris, you're probably not getting the amount of sanitation that you think. So you can't sanitize mud. I think that's how I boil this down. And Cheryl just put this link in the chat if you want to grab that. If you want other information, we've had a couple of publications on this. The one on the right was a Cal Ag article that kind of gives a lot of the overview of broomrape. We wrote this a couple of years ago, but it was kind of our first step in kind of getting a handle on this plant in California. There's a paper there where one of Matt's early projects was published. If you want to look at how the research progresses, those links are in the chat as well. This is kind of a media type article, but the UC Davis College of Agriculture and Environmental Sciences put together a nice overview of the work that UC Davis researchers are doing. A lot of what I talked about was presented in there, but also detection work, drone detection, volatile compound detection, breeding efforts, plant biologists. There's a lot of researchers that use a lot of research resources being brought to bear from the university, this university, as well as our cooperative extension and other university research collaborators. So this is kind of a neat article. There's some video in there. There's a lot of a nice overview of the project and the pest. So before I wrap up here, I want to acknowledge a few things. A lot of the funding for this came from the Tomato Research Institute, the USDA IR4 program, a fairly major grant from the California Department of Food and Agriculture Specialty Crop Block Grant. California League of Food Processors have funded some of the trailer work. And then some growers in the area have been super helpful with the field trial that I mentioned, as well as the harvesters, and just provided really good input on kind of the logistical challenges of some of the sanitation work in particular. And I've got Patricia and Jean, farm advisor and farm advisor emeritus from Yolo County, and Kobe Goldwasser is an Israeli researcher who's been super helpful in helping us get a handle on this problem. So I will stop there, and if I've got a minute or two for questions, go ahead and put them in the chat, or Cheryl, if you tell me what you want me to do.
So I'll go ahead and ask you a couple questions that came in. One of them is, what other plants do these weeds attack?
Yeah. Okay. So the broomrapes they attack a lot of different broadleaf plants. So in California, I've only seen it on tomato and on nightshades in a couple other fields, but the reported hosts include sunflower, safflower, cucurbits. There's a fairly broad host range. So there's some risk. Carrots, melons could be, but we have not seen it in California on those crops. So that's from the literature, but hemp is another one, but no grasses are in that host list, but a lot of the broadleaf crops are.
Okay, thank you. And then another one, are you using any adjuvants with the chemigation treatments?
No, that's just the straight herbicide in the chemigation water. Trying to run it over an hour or a couple hour period in the middle of an irrigation run to help with the distribution uniformity, but no adjuvants.
Okay, thank you. And another one, this one's from Daniel. What are the possibilities of broomrape of developing resistance to the chemicals being tested?
Unknown. I think there's always a risk of selecting for resistance, but I've never heard of resistance in broomrape in other countries. But it's kind of a niche weed problem, so it's not zero. But I think at this point, getting a control strategy is the main thing, and then we'll worry about the resistance issues down the road.
All right, and the next one's from Susan. What is the % infection to demand quarantine?
Zero detection. I mean, technically, if you have one broomrape plant, you're supposed to report it and quarantine that field. So there's not a threshold that you can live with. It's yes or no. That's what makes this such a problem. Most of the fields that I've seen that have had broomrape, you're talking about maybe a handful of plants. The field that we're working in is heavily infested, but the majority, it's just one or two here or there. So yeah, there's no threshold at this point.
Okay. And the next one is I don't know if I'm going to pronounce this correctly, is Amazamox registration off the table?
I think so. Amazamox is registered in alfalfa and some other crops here. I think the crop injury is too great. The efficacy was not, the efficacy was underwhelming, and the crop injury sometimes was minor, sometimes was major. I don't think we're going to see that registered. I'm not sure I would support registration of Amazamox. It's too iffy from a crop safety standpoint.
Okay, and then I think I'll just ask this last one, and then we'll go ahead and put the link to the test in the chat. So this last one, during the standard chemigation, which factor can improve or fail the herbicide application in the tomato crop?
If I understand the question correctly, the ways that I would see chemigation going poorly is injecting it in too short of a time, or so that you don't, so basically you get a hot spot of herbicide that doesn't distribute well in the rhizosphere where the tomato root is. So I think you want to inject it over a relatively long period of time with some water following it to push it out a little bit beyond, but you also don't want to run it too long and push the herbicide too far out of the root zone. So that's some of the things that I think we're still going to have to do research to understand. But in many ways, it'd be like thinking about your fertilizers. You don't want your nitrogen fertilizer to go beyond where the tomatoes can get it either. So it's the same basic idea.
Thank you so much for staying on and for your time and your presentation today.
Yeah, for sure, Cheryl.
All right, have a great afternoon.